Okay, so this lesson is going to have a look at more methodology within the biological psychology section. We're going to focus in this lesson on the role of correlations. Now, this is not only a process in psychological research, but it is quite important to understand the differences between correlational research, which can in fact draw on other methods. Things like questionnaires are quite often used in correlations, and also how this is extremely different from all of the experimental methods, in that it does not attempt to establish cause and effect, and you are only looking for relationships between two what we call covariables. So there's no manipulation of an IV, there's no DV that you measure. You're just looking at whether one thing affects another and finding out how or why that effect takes place, but you cannot make causal statements. So correlations are quite different to other forms of research methodology that we have had a look at. So what do you need to know about correlations? So we're going to have a look at the use of correlational research in psychology. So that is, what is a covariable, how are they measured, and so on. We'll look at the types of correlations, which positive, negative, and that's the use of scatter diagrams. You will have done scatter diagrams or scatter plots, as they may have been called at GCSE in maths. So this is something you should be familiar with. And then we're going to have a look at the issues around the use of correlations. So some of the issues with cause and effect, the influence of other variables, the fact that it is not really a scientific statement. You've got very little control, and unfortunately you can't manipulate any of those variables. A lot of correlations tend to be in retrospect, so asking somebody experiences of childhood and current behaviours and things like that. So the cause and effect issues are quite big when it comes to correlational research.
Hello everyone, and welcome to this short research methods lesson on correlations. Now, the way this is going to work is very simple. I'm going to give you a few little introductory points into what correlations are and how they work, but honestly a lot of it shouldn't be new to you because you will have come across it in other topics in psychology already. We're then going to move on to a few little examples and a few of the more nitty-gritty points before looking at some evaluation points and some possible exam questions. Okay, so correlations are a type of research method that illustrate the nature of an association between two covariables. Okay, so what that means essentially is that it looks at if there is a link or a relationship between two things. Okay, it looks at what that link is, and it also looks at how strong that link is. Typically, correlations are plotted on a scattergram. It looks a little bit like this. Okay, it's important that you know what this is because it's very common to ask people in an exam to draw a scattergram. When you're drawing a scattergram, you plot your covariables on each axis. So in this case, you are looking at the relationship between a wife's age and a husband's age. And as you can see, the wife's age gets plotted on the y-axis and the husband's age gets plotted on the x-axis. But it doesn't really matter. You could switch those around if you wanted to. Each one of the blue dots is a participant, or is a pair of participants in this case. Okay. The strength of a correlation is indicated by what's known as a correlation coefficient. Okay, so that's always designated by the letter R. And the correlation coefficient ranges from -1 to +1. Okay, -1 is a perfect negative correlation and +1 is a perfect positive correlation. And we'll come on to what that means in a sec. Okay, so like I said before, you have two types of correlation. You've got positive correlations and you have negative correlations. You've also got no correlation, but that's not really a type of correlation. So your positive correlation, you can see on the left-hand side there, that essentially is as one variable increases, so does the other. So for example, caffeine and anxiety, as the amount of caffeine that you drink increases, so does the level of anxiety that you experience. You've also got negative correlation, which means that as one variable increases, the other one decreases. So the example there would be that as the amount of caffeine that you drink increases, the amount that you sleep decreases. Okay, and then obviously no correlation means there's no relationship at all. In terms of your correlation coefficients, this is what I was saying before. So the positive correlation on the left there, you could say that that's a pretty much perfect correlation. So R would be +1, and the one in the middle, you've got a pretty much perfect negative correlation. So R would be -1, and for the no correlation, R would be 0. Okay, and R is the letter that we use for the correlation coefficient. So I've just got a few more examples for you there. Part of the mathematical requirement in A-level psychology is being able to estimate results. So it's not unheard of for students to be given a scattergram with the results of a correlational study and then being asked to estimate the correlation coefficient. Okay, so obviously in the previous slide you saw a pretty much perfect correlation, but they're not always going to be like that. So it's important that you realize what the difference is between a perfect correlation and, let's say, a correlation of 0.3 where it's far from perfect and the points on the scattergram are more spread out. Okay, so you've got 0.7 on the left, which is fairly decent but not perfect, 0.3 in the middle, which is quite spread out and quite far from perfect, and then 0 on the right where there's just nothing there. Okay, so being able to see the difference in those and make a judgment call as to how good and how strong the correlation is something that you're going to need to be able to do. Okay, so the final bit before we move on to the evaluation points is just making sure that everybody is clear on the difference between a correlation and an experiment, because a correlation is what's known as a non-experimental method. Experiments have IVs and DVs, and having an IV and a DV means that we can infer causality. Okay, so we can say the change in the DV was brought about by the manipulation of the IV. But correlations don't have any manipulation of variables. They don't have IVs and DVs, which means that it's not possible to infer any kind of causality. Because of that, you've always got to consider third-party variables. You've always got to consider intervening variables, variables that you're not studying but could have an effect on the variables that you are studying. An example of that comes from the social influence topic. So in the social influence topic, there is research support for the authoritarian personality, which was provided by Elms and Milgram. They found that people who scored highly on the F scale were also more likely to be obedient in Milgram's research. However, that finding is only correlational, because there are other variables that could have caused that level of obedience. For example, education levels. So there was also this argument that low levels of education could have also brought about high levels of obedience in Milgram's research. Okay, so the relationship between scores on the F scale and obedience in this case was only correlational, and there were other factors that had to be taken into consideration. Okay, correlation does not mean causation. Okay, there's always the possibility of an intervening variable. So that's the end of the outline already. We'll now move on to a couple of evaluation points. They're very, very short because, you know, this is a research methods topic, and so they don't tend to go into too much detail. Try and remember as many as you can. I can't see you ever needing more than 3 for any kind of evaluation question that comes up in a research methods section, but just to be safe, I would try and remember 2 strengths and 2 limitations. So we'll start with strengths. A good thing about correlations is that they are a good preliminary tool. Okay, so assessing the strength of a relationship, or the strength and more importantly the direction of a relationship, could suggest ideas for future research. Okay, so if you do a correlation and you find that there is a strong relationship between two variables, or your results throw up some kind of interesting pattern, then you might want to dig into that a little bit more. Okay, so correlations very often provide a good starting point for research, and people often conduct them before committing to long-term research. Okay, because you can do them quickly, and if there's nothing there, then you don't need to invest any more time. And then, like I just kind of alluded to, they are quick and easy to carry out. Okay, there's no need for a controlled environment like a lab. There's no manipulation of variables. It's simple. You could even use secondary data. So secondary data is data that's been collected by somebody else for a different purpose, like government statistics or government surveys and that kind of thing. You could even use those for your correlation. So you don't even need to collect your own data, which make correlational research even less time consuming. Okay, so it's very economical, it's very quick and easy to carry out. So moving on to limitations, there are 3 that I'm going to show you. The first one is the fact that it can only tell us how variables are related, but not why they're related. Okay, and that's due to the lack of experimental manipulation and control within a correlational study. Because they can't demonstrate cause and effect, we also can't be sure which variable is actually causing the other variable to change, which means we also have an issue with the direction of causality. So as an example, caffeine and anxiety are positively correlated. However, we don't know whether an increase in caffeine causes an increase in anxiety, or whether it's the fact that already anxious people tend to drink more coffee. Okay, so we don't know which of the two it is. Okay, so we've got a problem with the direction of causality. You've also got what's known as the third variable problem. We talked about this briefly earlier. So again, if we stick with the anxiety and caffeine issue, a third variable could be a high pressure job. So you could say somebody in a high pressure job experiences more anxiety and therefore drinks more caffeine. Or you could say somebody in a high pressure job drinks more caffeine to keep up with the long working hours, and because they are drinking more caffeine, they're also experiencing more anxiety. Okay, so the third variable of a high pressure job could be the important variable and could be the one that's affecting both caffeine and anxiety, but we don't know. Okay, so there's always this issue of an intervening variable. And then finally, you've got the problem that correlational research is very often misused and misinterpreted. Okay, so remember, correlational research only shows us relationships, okay, and whether or not they exist and how strong they are. Very often these relationships, however, are misquoted as facts, and they're not facts, they're just associations. An example of that is research that found that people who grew up in single-parent households are more likely to go on and commit crime. Okay, now that doesn't mean that growing up in a single-parent household makes you a criminal, because there's a lot of other third-party variables and intervening variables that exist there. Okay, however, this relationship has in the past been misquoted as a fact when it's not. It's just a relationship. Okay, so the results can very often be misquoted and misinterpreted, which is an issue. Okay, just to finish off then, I've got a couple
So hopefully that overview gave you a little bit of information. Don't necessarily focus on the exam skills advice there. It was possibly not in relation to the nature of the question that you may well get in Edexcel A-level psychology. I would certainly suggest that you need 4 strengths, 4 weaknesses, and that you need to support those with links to correlational research that's taken place predominantly across biological psychology, but you can draw on other aspects as well. So, given that there are a number of issues with correlational research methods, the question is obviously why would researchers embark on investigating correlations? Okay, so one of the first reasons is that if researchers are interested in finding out whether there is a statistical relationship between particular variables, they may conduct a correlational study as a pilot study or a preliminary study, rather than investing in the time and depth that an experiment would require, only for them to then find out that they've tested the wrong variables. So it may be that they look at correlations first of all to see if there is an initial relationship between different covariables, to establish whether that is either a positive, negative relationship, if it's a strong enough relationship that is worth testing, prior to then conducting more experimental or structured observational methods to start to try and establish cause and effect around that relationship. Secondly, if they find that the variables are related to each other, obviously it's very difficult in that sense to determine which causes the other. I mean, the example in the video talks about caffeine, anxiety, and also high pressure job. It could also be, a lack of time to relax. It could be work pressure, financial pressure, all sorts of different things. So in order to look for that relationship, you would need to do multiple correlations to find out which of those covariables you would want to look further into if you are looking at conducting experiments. So whilst you can do multiple tests of covariables, you do need to remember that neither of those covariables are at this point an independent or dependent variable. They just simply do not apply to this kind of research. You are just looking at what kind of goes on, getting a picture, and then you would perhaps look in more detail. So choosing to use a correlational study rather than an experiment is to provide that sort of level of interest in a particular area. It might be hours spent revising and exam scores. It might be the hours that you sleep compared to physical energy that you have during a day. It's also very useful in research where you cannot manipulate a variable due to serious ethical issues or the fact that it is impossible or impractical. So things like preventing people from sleeping or making people take recreational drugs, to look at whether or not there's a relationship between that and particular aspects of behaviour, would be considered far too unethical to be passed by the BPS Code of Ethics Board. So the fact that covariables are pre-existing is something that allows you to kind of get past some of those ethical issues of manipulating variables that could cause serious long-term harm or damage to participants. So when you conduct a correlation, you present your data first of all on a scatter plot, and this will show you visually one of the 3 main types of correlation. So you've got a positive correlation. So this is when one score rises, the other score rises. So for example, as age increases, so as you get older, the time it takes to react to a stimulus also gets longer. So time goes up as age goes up. That would be known as a positive correlation. Negative correlation is when one score rises as the other falls. So, for example, as age increases, average driving speed falls. So there's a negative correlation. The older you are, the slower you tend to drive. And then finally, zero correlation is when there's no relationship at all between the variables. So on a scatter diagram, all your little dots and crosses would be all over the place, showing absolutely no relationship whatsoever between different variables. So, quickfire recap, there is no IV or DV, there are just covariables and they're of equal importance. The hypothesis that you'll be testing is not about difference, so we're not looking at a difference between or whether something is more or less. It will be about a relationship, so whether there is a relationship between caffeine and anxiety. The hypothesis can be directional because it can predict a positive or negative correlational relationship. So there is a positive relationship between anxiety and caffeine, or there is a negative relationship between anxiety and caffeine, and things like that. So you can predict in a directional hypothesis which direction your correlation will be, positive or negative. But you must remember, in any correlational hypothesis, you absolutely have to be talking about relationships between covariables and positive or negative direction, and there is no other kind of relationship directional statement that you can make. A scatter diagram shows you visually whether the correlation is positive, negative, or neither. Correlations, like all other research methods, involve the same sampling techniques, so you would still use your random, stratified, opportunity or volunteer sampling techniques within it. However, they do not have the same design as any of the other methods that you study in psychology. So they don't involve randomizing people into groups because it's a within participant design. So each participant is scored for each of the two covariables. There's no need to sort people out into groups. You don't need to have independent groups because you're testing the same people. It's not repeated measures because you are not testing them on two different versions of an IV, right? So it is known as within participant design. And because this is the only thing that you actually have to use that for, it's not an experimental design that you would be asked directly on in an exam anyway. All right, so you just need to remember that it does not have one of the experimental research designs that you have learned about. It's within participant design, so you don't need to worry about anything like individual differences and so on, because they are scored on both of the conditions, so both of the covariables. So if you have a look in your booklet, you can see an example by Matthews et al. In 2012. What I'd like you to do is just read through that example as it gives an idea of how correlation would be completed. This correlation in particular uses MRI scanning and lifetime aggression scores. So remember, when a correlation is done, it uses another research method to gather the data on the covariables. So a correlation in itself is the process by which you discover relationships. It is not a fixed specific type of method. You would embed into it something like a questionnaire, or in this case an MRI scan, or data from an observation, things like that. So have a read through the example in your booklets, highlight the key points and make notes, and then restart the lesson when you're done reading Matthews' study. Okay, so some of the strengths of using a correlation. It is very, very, very beneficial to find out those initial relationships. They are things that may not have been realised prior to a correlation being conducted. So it highlights some of those new ideas. It gives you ideas to generate further research. It allows you to look at a whole host of different things and then begin to develop that into the finer points of more experimental type research methods. Secondly, because the same people are providing both sets of data and it's a within participant design, it will not be affected by individual differences in that sense. So the results are not affected by participant variables. You've got the same people doing both of the covariables, so being scored on both of them. What you do have to bear in mind is that whilst there are no participant variables, like the video talked about and I mentioned earlier, there is the third variable issue or other covariables that you haven't taken into account that could skew the data. But participant variables are not one of the weaknesses in a correlation because it is the same people that you are looking at. Okay, so you need to add the following strengths to your booklet as well. So it allows researchers to determine the strength and direction of a relationship, so later studies can narrow the findings down and, if possible, determine causation experimentally. So if you found out there was a relationship between caffeine and anxiety, for example, you may then want to undertake further research looking at the implications of caffeine, maybe testing the effect of caffeine on anxiety levels in things like rats. You wouldn't be able to do that in humans because of ethical issues. You couldn't create anxiety by giving people too much caffeine. That would be woefully unethical, but you could do it with animal research. The second one to add, because correlational research usually takes place outside the lab, the results tend to be more applicable to everyday life. So if you are asking somebody, how much coffee do you drink? They're just going to tell you how much coffee you drink. The only time this gets a little bit problematic is if you're doing things like prejudice, obedience, aggression, and there is the social desirability issue of the questionnaire or the interview or however you're gathering your data that could skew the way in which they respond, giving more socially desirable answers. So that element does play a part in sort of the nature of correlation because it obviously influences the research method that you are using to gather your correlational data. But ultimately, an awful lot of correlation does ask about day-to-day situations where you can draw on real-life experiences and allow people to give their own answers and their own scores for things like that. So if you've not copied those down, just pause the video and then restart when you've finished. So looking at the weaknesses of correlation, it only indicates the relationship, remember, between the two covariables. So there is absolutely no cause and effect identified, as there's no IV or DV. You've got no IV to manipulate, no DV to measure. So because of that, you cannot establish whether one affects the other. Now, this lack of cause and effect makes correlation less scientific. So you have less scientific data and therefore you can't establish those scientific principles or falsify correlations to support theories and ideas in a scientific manner. The measures themselves might not provide valid data, so it's very difficult to sometimes quantify some of the things you would want to measure in a correlation. And again, this goes back to those ideas of issues with questionnaires and things like that. You can count the number of hours somebody spent in therapy, but quantifying the benefits of therapy to see if, you know, the longer you spend in therapy, the better you feel. How do you score somebody's benefit of therapy? How do you score somebody's happiness level? How do you score somebody's ability to feel good about themselves? One person may score themselves as a 10, another person may score themselves as a 5, but in actual fact, their feelings may be exactly the same. They just expect more of themselves and things like that. So some of the measures of covariables can be very, very difficult to test objectively. Finally, if you add the last weakness here, this goes back to the idea of that third variable or the unknown variable that may be causing the relationships between the covariables that you're testing to actually show as some sort of positive or negative correlation. And it's good to go back to that video idea of high pressure job, caffeine, anxiety. It's called the third variable issue, but actually there could be multiple variables. It could be a high pressure job. It could be financial responsibility. It could be a lack of time at home. It could be a lack of time engaging in leisure to bring your anxiety levels down. So those issues around the covariables cannot be controlled, and you will never really know what the actual relationship is coming from. So it can make it quite difficult to test sort of clearly relationships between covariables. You would actually have to do several correlational studies in that case to discover whether caffeine and anxiety link, or whether anxiety and high pressure jobs link, and things like that. So you'd have to do multiple correlations in that respect. So we're going to have a look at the ways in which you analyse correlational data, and that includes scatter diagrams and the use of a Spearman's test. Now, the Spearman's test we're going to do in the biological practical, so I'm not going to dwell on that towards the end too much, but we're going to have a look first of all at scatter diagrams. So before we go into the detail of scatter diagrams, one of the things that you do need to be aware of is what we call sense checking data. Now, you need to be able to analyse and interpret patterns from data and carry out what we call sense checks. So that's literally just having a look, looking at your data. Do you see a pattern? Is there a pattern? Is it worth going into this in a little bit more detail? Does it look significantly related? So this is taught at this point because it's quite easy to do this with a scatter diagram, because literally you just look at where the dots appear on the scatter plot of the two covariables. And that's what we mean by sense checking data. Having a look at your numbers and thinking, does it look like I've got anything of value here? You also sense check all forms of data. So for example, if you think back to your cognitive practical, where we were looking at a difference between recall in a 3-second and an 18-second interference task, if you looked at that data and everyone scored the same number of trigrams, so they all recalled 3 in the 3-second interference and they all recalled 3 in the 18-second interference, it's fairly easy to look at the fact that everyone scored exactly the same in all the conditions to realise that actually something's gone wrong. That data doesn't have any differences between it. It doesn't quite look right. You might want to redo the experiment, or you might want to look at a different way of undertaking that experiment. So you use sense checking across all forms of data, not just correlations and scatter diagrams. So correlational data displayed in a scatter diagram is something you will be familiar with from GCSE maths. It's often called scatter diagram, scatter graph, scattergram, scatter plot. Whatever name is given to it is fine. It is the same way of presenting data. So the 2 scores from each participant on the covariables are drawn together to generate a point on the graph. A line of best fit can be drawn through the middle to see how far those scattered plots of covariable scores actually fit in terms of their closeness to the correlational line. So you can see in the images there in front of you, you've got 3 different types of correlation shown. Now, you've got your first one, which is your positive correlation. Now, this pretty much shows a perfect positive correlation. Quite often, this is not what you'll get. There'll be a little bit more spread out. But to sense check that, you would look at that data and go, okay, looks like there's a positive relationship there. Can be quite clear with that. And you would go on to then statistically test the correlation data that you've gathered. The next one is a negative correlation. Again, this one shows a pretty much perfect negative correlation. So again, on a sense check, you would look at that and go, yeah, that looks pretty much like they're all around that line of best fit. So I would say there is some sort of relationship, and it's clearly a negative relationship between the covariables. The final one is no correlation. So on this one, if you did draw a line of best fit, which would be something like that, or something like that, if you're going for negatives, you would see that they are not anywhere near the line of best fit. So when looking at that particular data, you're able to sort of sense check and go, actually, these are dotted around all over the place. There's nothing consistent there, and therefore there is no relationship between the data that I have gathered. So that's how you would go about sense checking those kinds of data. Another thing that is worth bearing in mind with correlations, it's very easy to spot anomalies in your data. Now, I know I've talked about anomalies quite a lot when we talk about the effect of anomalies on results. So if you have a participant who scores really weirdly in one of your experiments or observations, or correlation in this case, the effect of anomalies on the data is linked to your reliability. So this is where we're talking about sample size. So where you have a very large sample and you've got an anomalous result, so let's put Bob. All right, Bob scores here. All right, that little red dot. Bob is an anomaly. All right, the vast majority of data is here. So what you can do with the correlation as well is you can spot outliers, and they're the ones that lie really far outside of your normal sort of distribution of correlational data. All right, so you've got Bob up here, don't know what happened to him, so we're going to ignore Bob's data. All right, we're going to go, Bob, you're an outlier, off you go. Something wrong with your responses, and so on. So that's what we're talking about when we're talking about anomalies in the data. Obviously, if you've got a small sample, the more anomalies you have, the worse the effect may be on something like your correlations. So you've got like all these anomalies over here. All right, and that's where it starts to cause a problem. All right, so sense checking that data, actually you're going to question whether your covariable matches your other covariable along here. All right, so the bigger the sample you have, the more condensed your data is going to be, and therefore the more reliable your test, in this case of covariables, but in an experiment of your IV and DV, going to very clearly, look at all those dots, all right, bigger sample, right, the effect of these outliers is much less on the data that you've got. So that helps put that, I hope, into context as well. Okay, so recap, positive correlation, right? This is where your line of best fit goes in this direction. All right, so your covariables plotted here and here, right? Up they go. All right, and it shows a positive relationship as this one goes up in number, this one goes up in number. The second one is your negative correlation. So again, you've got your covariable and your other covariable. This is where your data comes downwards. All right, so as one covariable increases, all right, the other variable decreases. So for example, if you've got age going up along the bottom here, your score for speed, for example, is coming down on that axis. All right, so that's your negative correlation. And like I said, no correlation at all, right, is where you've got your dots just all over the place, showing that there's absolutely nothing going on between this covariable and this covariable. So this brings us to statistical testing. Now the statistical test done for a correlation is a Spearman's rank correlation coefficient. And this is carried out to see if the relationship for the group is statistically significant. So whilst we may see that there's a positive correlation or a negative correlation, we don't know how close that correlation is, how close that relationship is, until we've actually done a statistical test. Now, this statistical test is referred to on a scale of +1 to -1. So +1, right, relates to positive. All right, so the plus sign, positive. All right, remember it's a mathematical term. All right, so you've got your positive direction indicated by a plus sign. Your -1, which is your perfect negative correlation, again, minus being negative. All right, so it goes along a scale. All right, you'll have your +1, you'll have your -1, you'll have your 0. Your correlation coefficient will work out score. So your score might be, what's this, 0.7, right? So that would say that you are statistically significant in that you've got a positive correlation. It's not quite perfect, but you know, it's all right. All right. Whereas here you may have a negative correlation of -4, right? And in that case, you know that your direction is negative, but your correlation is 0.4. Now your aim is to hit 1. So actually 0.4 is quite a long way from your goal. All right, that's quite a distance. It's closer to 0, and actually 0 would suggest there's absolutely no relationship going on whatsoever, and therefore this result here is actually quite a poor correlation. All right, so the closer to 1 or -1 your result, the better your correlation is. All right, the closer to 0, then the worse your correlation is, and the least significant your data becomes. So the formula and tables are provided in your exam. There is a table given to work out this, all right, to a certain extent. However, this is one of the few formulas where you are actually going to have to do a little bit of substitution into the formula. So this will be covered in your biological practical, and then we can do some recap on how you actually work the formulaic response out for a Spearman's correlation. So don't worry about that too much. You do not, under any circumstances, need to remember this. All right, you do not try and remember this formula. It is given to you. You just need to remember how to use it. So the reasons when you would use a Spearman's. All right, so key things. You use Spearman's rank, and this is the only SATs test for a correlation, right? So if you have a test of a relationship between two covariables, it's a Spearman's. If you have a within participant design, so that is the same participant filling out the two different versions of the covariable scores, you use a Spearman's. And the data is at least ordinal level, right? It's called a Spearman's rank test. You have to rank, order the data. So the data is ordinal. All right, so that's your third tick to make sure that you use a Spearman's test and not any others. It's quite easy to know when to do a Spearman's. If you are looking at any correlation study, it's a Spearman's. It's the only correlational statistical test that you learn in this course. So if it's a correlation, if it's looking for relationships, it will be a Spearman's test. Okay, so we don't want to dwell on this too much because we will do it as a class, but the Spearman's correlation coefficient is given as a little r, all right? And as I said before, the values go from +1 to -1, with +1 a perfect association in a positive direction, 0 no association, and -1 a perfect negative association. The closer you get to 0, the weaker your results are. All right, so you would be making statements if you had a score of 0.987, all right, that would indicate a very strong positive correlation, right? If you had a score of 0.2, that would indicate it's an extremely weak positive correlation, and probably not really any relationship significance between the two covariables. So this is the table that you would get in your exam paper. This is the one we will work through when we do your biological practical. But just to point out some of the key things. So you would be given this column, this column, this column, and this column. All right, so you're given the ranks. You then need to work out the difference. So as you can see, 6.5 take away 6 equals minus 0.5. All right. This one's obviously been done the other way, actually, 6 minus 6.5, because it's a minus 0.5. Don't worry which way you do it. So you can do 6 take away 6.5 or 6.5 take away 6. It makes no difference whatsoever. So you would be expected to work out this column first of all. Right, you go through all of those, all right, and that's 1 mark. You would then be expected to work out the difference squared. So this is just literally squaring the result that you've got here. Right, press the button on your calculator, squares. All right, work out each of those. Once you've done that, that's that mark ticked. All right, that takes you then to this part of your formula up at the top. So you would need to do 6 times the sum of d squared. So basically you at that point add all these together, all right, all the way to the end, and work out what the total is. All right, once you've got that total, times it by 6, right, and that's the bit that goes on your top line there. Okay, so for this data, that total is 12 times 6. Gosh, it's really hard to write on this. Which comes to 72. All right, so that's your top bit. All right, so 6 times the sum of d squared. All right, the sum of this is 12 times 6 is 72. We then need to work out the bottom section. So n is always the number of participants. So 1, 2, 3, 4, 5, 6, 7. So n is 7. We've got 7 people. Okay, so n squared is the bit in brackets. So it's 7 times 7 squared, which is 7 sevens are 49, minus 1. All right, so that translates to 7 times 48. All right, so we work that out. 7 times 48 is 336. Okay, so that's worked out the whole of this bit here. Now the bit that everybody forgets on this is the starting point, which is 1 minus. Right, so don't forget that little bit there. So we've got 1 minus, and then we need to work out 72 divided by 336, which gives you 0.214. Okay, so 1 minus 0.214 is, let's do your answer in a different colour here, 0.786. And there you go. That is your Spearman's rank correlation coefficient for the data on the screen. It's a positive number, right? So we know that we've got a positive correlation. 0.786, if you think of it on that scale of 0 to 1, is around here. So it's quite a strong positive correlation. It's not a very strong positive correlation. You'd expect that to be sort of in this region here, sort of 8 and above, or 8.5 and above. But here you've got quite a strong correlation, right? So for this question, we're looking at a strong positive correlation between self-rated aggression and peer-rated aggression. So in this case, people generally agree with their friends as to how aggressive they actually are. Right, so that's the basic principle of the Spearman's rank correlation coefficient. Don't worry too much about it. If you want to go back over it, you can do, but ultimately we will do this again for your biological practical. So if you do want a little bit of practice yourself, you can have another go at this example here. So you've got math scores and English. So you've got your rank, your difference between your ranks, and the difference squared. So that bit's all done for you. So this is basically substituting into the equation and having a go at working it out. So if you do want to have a little go at it, you can do. If you're looking at that thinking, nope, got no idea, don't worry, we will go over it in your biological practical anyway. Okay, so some of the things to recap on at this point. We will do more on your stats test, but you might want to have a look at probability levels and levels of significance, which we talked about mostly in cognitive psychology. You may want to have a quick look at type 1, type 2 errors, where you accept or reject your hypothesis incorrectly as a result of an incorrect calculation using the wrong level of significance, and what we mean by levels of significance, so understanding the tables at the front of an exam paper. Recap on the use of different types of hypotheses, so alternate includes correlational, but also look back at experimental and null hypotheses in cognitive. We've looked here at the use of covariables and correlations, but it's worth recapping on what an IV and a DV actually is in an experiment and how they're different so that you remember the differences between them. And again, go back to some of the main methodological issues that go through a range of methods. So the use of control groups, again, independent groups design in particular, having a baseline measure, things like Badley and Milgram for that. The use of control groups in RAIN, so testing brain scanning on NGRI murderers and also a control group. Have a look back at how you allocate people to groups, the randomization of groups. I know you don't need to do that for correlation because it's all within participant design, but you do need to recap on how you allocate people into those groups, the randomizing of your participants, and the randomizing of your conditions using things like ABBA and so on. And also recap on your sampling techniques, volunteer, opportunity, stratified and random sampling. So that would be handy to go away and just make sure that you're okay with all those key terms across your research methods. Any questions, pop a message on Teams or drop me an email. And if you do want to have a go at the Spearman's, by all means, have a go. And if you don't, feel free to just leave it until we do your biological practical.